Adenylyl cyclase, EC 4.6.1.1. Also commonly known as adenyl cyclase and adenylate cyclase, abbreviated AC, is an enzyme with key regulatory roles in essentially all cells. It is the most polyphyletic known enzyme. 6 distinct classes have been described, all catalyzing the same reaction but representing unrelated gene families with no known sequence or structural homology. The best-known class of adenylyl cyclases is class 3 or AC3. Roman numerals are used for classes. AC3 occurs widely in eukaryotes and has important roles in many human tissues. All classes of adenylyl cyclases catalyze the conversion of adenosine triphosphate, ATP, to 3'5 cyclic camp, and pyrophosphate. Magnesium ions are generally required and appears to be closely involved in the enzymatic mechanism. The camp produced by AC then serves as a regulatory signal via specific camp-binding proteins, either transcription factors, enzymes, e.g., camp-dependent kinases, or ion transporters. Classes. Class 1. The first class of adenylyl cyclases occur in many bacteria including E. coli. This was the first class of AC to be characterized. It was observed that E. coli deprived of glucose produce camp that serves as an internal signal to activate expression of genes for importing and metabolizing other sugars. Camp exerts this effect by binding the transcription factor CRP, also known as CAP. Class 1 ACs are large cytosolic enzymes, approximately 100 kDa, with a large regulatory domain, approximately 50 kDa, that indirectly senses glucose levels. As of 2012, no crystal structure is available for Class 1 AC. Class 2. These adenylyl cyclases are toxins secreted by pathogenic bacteria such as Bacillus anthracis and Bordetella pertussis during infection. These bacteria also secrete proteins that enable the AC2 to enter host cells, where the exogenous AC activity undermines normal cellular processes. The genes for Class 2 ACs are known as a CYAA. Several crystal structures are known for AC2 enzymes. Class 3. These adenylyl cyclases are the most familiar based on extensive study due to their important roles in human health. They are also found in some bacteria, notably Mycobacterium tuberculosis, where they appear to have a key role in pathogenesis. Most ACIIs are integral membrane proteins involved in transducing extracellular signals into intracellular responses. A Nobel Prize was awarded to Earl Sutherland in 1971 for discovering the key role of AC3 in human liver, where adrenaline indirectly stimulates AC to mobilize stored energy in the fight-or-flight response. The effect of adrenaline is via a G protein signaling cascade, which transmits chemical signals from outside the cell across the membrane to the inside of the cell, cytoplasm. The outside signal, in this case, adrenaline, binds to a receptor, which transmits a signal to the G protein, which transmits a signal to adenylyl cyclase, which transmits a signal by converting adenosine triphosphate to cyclic adenosine monophosphate. CAMP is known as a second messenger. Cyclic AMP is an important molecule in eukaryotic signal transduction. A so-called second messenger. Adenylyl cyclases are often activated or inhibited by G proteins, which are coupled to membrane receptors and thus can respond to hormonal or other stimuli. Following activation of adenylyl cyclase, the resulting CAMP acts as a second messenger by interacting with and regulating other proteins, such as protein kinase A and cyclic nucleotide-gated ion channels. Photoactivatable adenylyl cyclase, PAC, was discovered in E. gratulus and can be expressed in other organisms through genetic manipulation. Shining blue light on a cell containing PAC activates it and abruptly increases the rate of conversion of ATP to CAMP. This is a useful technique for researchers in neuroscience because it allows them to quickly increase the intracellular CAMP levels in particular neurons, and to study the effect of that increase in neural activity on the behavior of the organism. For example, PAC expression in certain neurons has been shown to alter the grooming behavior in fruit flies exposed to blue light. Chanelrhodopsin-2 is also used in a similar fashion. Structure. Most class 3 adenylyl cyclases are transmembrane proteins with 12 transmembrane segments. The protein is organized with 6 transmembrane segments, then the C1 cytoplasmic domain, then another 6 membrane segments, and then a second cytoplasmic domain called C2. The important parts for function are the N-terminus and the C1 and C2 regions. The C1a and C2a subdomains are homologous and form an intramolecular dimer that forms the active site. In Mycobacterium tuberculosis, the AC3 polypeptide is only half as long, comprising one 6-transmembrane dimer followed by a cytoplasmic domain. But 2 of these form a functional homodimer that resembles the mammalian architecture. Types. There are 10 known isoforms of adenylyl cyclases in mammals. These are also sometimes called simply AC1, AC2, etc., and, somewhat confusingly, sometimes Roman numerals are used for these isoforms that all belong to the overall AC class 3. They differ mainly in how they are regulated, and are differentially expressed in various tissues throughout mammalian development. Regulation. Adenylyl cyclase is dually regulated by G proteins, G's stimulating activity and G inhibiting it, and by forskolin, as well as other isoform-specific effectors. Isoforms 3, V and 8 are also stimulated by circa 2+ kilmodulin. Isoforms I and Vi are inhibited by circa 2+ in a kilmodulin-independent manner. Isoforms 2, IV and IX are stimulated by beta-gamma subunits of the G protein. Isoforms I, V and Vi are most clearly inhibited by G, while other isoforms show less dual regulation by the inhibitory G protein. Soluble AC, SAC, is not a transmembrane form and is not regulated by G proteins or forskolin. Instead acts as a bicarbonate pH sensor. It is anchored at various locations within the cell and, with phosphodiesterases, forms local CAMP signaling domains. In neurons, calcium-sensitive adenylyl cyclases are located next to calcium ion channels for faster reaction to circa 2+ influx. They are suspected of playing an important role in learning processes. This is supported by the fact that adenylyl cyclases are coincidence detectors, meaning that they are activated only by several different signals occurring together. Source: in peripheral cells and tissues, adenylyl cyclases appear to form molecular complexes with specific receptors and other signaling proteins in an isoform-specific manner. Class 4 ACIV was first reported in the bacterium Aeromonas hydrophila, and the structure of the ACIV from Yersinia pestis has been reported. These are the smallest of the AC enzyme classes. The ACIV from Yersinia is a dimer of 19 kDa subunits with no known regulatory components. Classes V and Vi. These forms of AC have been reported in specific bacteria, Prevotella ruminicola and Rhizobium eti, respectively, and have not been extensively characterized. Additional images. Further reading. Adenylyl cyclase at the U.S. National Library of Medicine. Medical subject headings. MESH. Interactive 3D views of adenylate cyclase at Proteopedia. Adenylyl _cyclase